Welcome to today's episode, where we dive deep into one of the most remarkable explorations of human nature ever written. Behave, the biology of humans at our best and worst, by legendary neuroscientist and primatologist Robert Sapolsky.
Welcome to Fact Horizon. This is where history meets mystery and facts define the future. We explore the profound truths of human existence, the wonders of the mind, and the secrets of the cosmos, all in one place. If you seek knowledge and wonder, you're in the right spot. Subscribe now and join our journey beyond the horizon.
And we are just so incredibly thrilled you're with us for this deep dive today because we're basically tearing down this whole illusion of the independent self. We're tackling the biggest, arguably the most haunting question there is.
Right, the classic, why do we do the things we do? And specifically, why do we humans harm one another so deeply? And, why do we do the exact opposite?
Exactly. And Robert Sapolsky's book, Behave, it just forces us to look in the mirror. I mean, it is this exhaustive, incredible investigation into the biology of our actions. But right out of the gate, he insists that to understand behavior, we have to completely destroy how we currently think about it.
Yeah, he takes a sledgehammer to it. We have to reject what he calls categorical thinking. And, that rejection, it's the absolute bedrock of this entire exploration.
So let's unpack that. Categorical thinking, that's just our habit of putting things in neat little boxes, right?
Very much, yeah. It's a deeply ingrained cognitive habit for us. It's how human brains are wired to handle overwhelming complexity. When we face something really messy, like violence or compassion, we try to isolate the explanations.
Yeah.
Like saying, oh, that behavior is just genetics, or it's entirely because of their childhood trauma. Right. Or it's just an evolutionary adaptation. We try to find this single scientific discipline to explain a massive phenomenon. But Sapolsky argues that artificially forcing facts into these cleanly demarcated buckets, it completely destroys our ability to understand the actual mechanism.
Because the boundaries between those disciplines like neurobiology and endocrinology, they're just human inventions. They're totally arbitrary.
Exactly. The moment you invoke a neurobiological explanation, you are implicitly invoking the environment that shaped that neurobiology and the hormones that primed it and, the evolutionary history that literally designed it.
Okay, so I like to think of this like trying to understand a massive symphony by only listening to the tuba. I mean, if you only look through the lens of one discipline, you completely miss the music.
That is a perfect analogy. You miss the music entirely.
Right. You might know exactly what the tuba is doing mechanically, but you have zero idea what the violins are doing or the percussion or how the conductor is guiding the overall sound. You miss the context.
You miss the harmony, the dissonance, all of it. If you say a behavior is caused by a gene, well, you're missing the fact that the environment influenced how that gene was expressed in the first place.
And you're missing the evolutionary pressures from millions of years ago that shaped why that gene even exists. But the danger here isn't just academic, is it?
Oh, not at all. It's catastrophic. History is literally littered with the wreckage of brilliant scientists who, they fell into the trap of the tuba. Sapolsky provides these chilling historical examples of influential minds getting stuck in a single bucket.
Like John Watson, right, in the 1920s. The behavioralism guy.
Yes, John Watson. He famously claimed that if you gave him a dozen healthy infants and total control over their world, he could train any one of them to become any type of specialist, a doctor, a lawyer, even a beggarman thief.
Regardless of their innate talents or ancestry, he just completely ignored biology.
Totally ignored it. He was so deeply trapped in the environmental bucket that he erased the vast biological diversity in human potential. Or, look at Egas Moniz.
Wait, the guy who invented the lobotomy?
That's him. He was a neurologist who actually won a Nobel Prize for developing the frontal lobotomy. He was mired in this crude, isolated neurological bucket. He genuinely believed complex mental disorders could be cured by just severing synapses with an ice pick.
That is just horrifying. And he won a Nobel Prize for it.
He did. And then there's Konrad Lorenz, the brilliant animal behaviorist. He actually became a Nazi propagandist. Lorenz was pathologically stuck in a gross misinterpretation of the genetic bucket. He believed society needed to actively select for toughness to avoid biological degeneracy.
Wow. So when we rely on a single disciplinary bucket, we aren't just misunderstanding the mechanics, we risk endorsing these catastrophic social policies.
Exactly. It changes how we treat human beings on a massive scale.
Those examples really ground the stakes here. But, I mean, if we throw away these isolated buckets, how do we map this out without getting totally overwhelmed by the complexity? What's Sapolsky's alternative?
The alternative is a chronological synthesis. We have to view human behavior through a continuous backward-looking timeline. So let's say a behavior happens. A human physically moves their muscles.
Okay, they throw a punch, or they reach out to hold someone's hand.
Right. To understand why, we start by looking exactly 1 second before the action. That puts us in neurobiology. What was the physical architecture of the brain doing just a second prior? Then we pull the camera back further.
To what? The sensory stuff.
Yes, the sensory stimuli in the seconds to minutes before that triggered the nervous system. Then we pull back to hours or days before. What hormones altered the brain's sensitivity to those stimuli?
So we keep expanding the timeline, weeks and months before, looking at neuroplasticity, then years back to childhood and even fetal development in the womb.
And we don't stop there. We go back to the individual's genetic makeup. Then we zoom out to the culture that shaped their group, the ecological factors shaping that culture, and finally, millennia back to evolutionary pressures. Every single level of explanation is just the end product of the influences that preceded it.
This is part 1 of our comprehensive series on Behave. In this first part, we will explore the foundational biological mechanisms occurring seconds to minutes before a behavior, specifically breaking down the concepts in the following chapters. Chapter 1, the behavior. Chapter 2, 1 second before.
And as we navigate Chapter 1, the behavior, we immediately hit a massive definitional roadblock.
Defining the behavior itself, right, because words like aggression or altruism are surprisingly tricky.
They're a nightmare to define biologically. And the reason is that the physical motor action, the actual flexing of muscles and movement of bone, it tells us almost nothing. The meaning of any action is intrinsically dependent on context.
Right, the physical action is completely ambiguous. Think about the act of pulling a trigger. I mean, pulling a trigger could be an act of hideous, cold-blooded murder.
Absolutely.
But pulling a trigger could also be a desperate act of heroic self-sacrifice, like drawing enemy fire to save your platoon. The exact same muscles in the forearm contract. The exact same joints in the finger flex.
But the moral meaning is completely inverted based on the external context. We do not hate violence inherently. We hate the wrong kind of violence. We hate violence in the wrong context.
But violence in the right context, we love it. We cheer for it in stadiums. We hand out medals for it.
We build statues for it.
Mm-hmm.
And the book details this absolutely fascinating fMRI study that perfectly illustrates how deeply the brain cares about context over motor movement. Researchers put subjects in a brain scanner and immerse them in a virtual reality simulation.
Okay, so what did they have them do in the VR?
In one scenario, they encountered an injured person in need of help, and they had to perform the physical motion of applying a bandage. In another scenario, they encountered a menacing extraterrestrial, and they had to perform the physical motion of shooting a gun.
And obviously, bandaging an arm and pulling a trigger are totally different physical actions. They require entirely different motor cortex mapping.
Completely different on a biomechanical level. Yet, when researchers looked at the subject's prefrontal cortices, the part managing complex decisions, the contemplation of both of those actions activated the exact same circuitry.
Wait, really? The exact same neural circuits for shooting an alien and bandaging a wound?
The exact same.
What?
Why? Because in the context of that virtual room, both actions represented the identical abstract concept. They both meant doing the right thing.
Wow. So the brain cares way less about the mechanics of the muscle and infinitely more about the appropriateness of the action.
Exactly. The biological mechanisms are about the deep contextual meaning behind the action, which makes pinning down a biological signature for something like aggression incredibly complicated. Because aggression isn't a monolith.
No, there's a real duality to it. We intuitively understand the difference between someone losing their temper and throwing a punch in a bar, which is, reactive, hot-blooded aggression.
Right, impulsive.
Versus a hitman methodically assembling a sniper rifle from a mile away. That's instrumental, cold-blooded aggression.
And separating those two flavors is crucial because the underlying biology is vastly different. But one of the most pervasive and frankly troubling forms of aggression across all species is something called displacement aggression.
Displacement aggression. Let's dig into that.
This happens when an individual is stressed, frustrated, or in pain, and they displace that negative physiological state onto a third innocent party. If you take a rat and deliver mild electrical shocks to it, it is highly likely to bite a smaller innocent rat placed nearby.
Just to take it out on them.
Exactly. In primate troops, if a high-ranking alpha male baboon loses a fight to a rival, he won't just sit there. He will often immediately chase down a subordinate male and attack him.
And let me guess, that subordinate just passes it down the line.
Yes. The subordinate turns around and bites a female, who then lunges at an infant. It becomes this cascading chain of hostility down the social ladder.
I need to understand the mechanism there. Why on earth does the brain do that? It seems like a massive waste of energy. If you're stressed or injured, shouldn't evolution program you to conserve your resources, not pick a random fight with a bystander?
You would think so, but the depressing biological reality lies in our stress response. When you experience a stressor, your body floods with glucocorticoids, like cortisol. Your allostatic load skyrockets.
So your heart rate goes up, your blood pressure spikes.
Your system is screaming for a way to restore homeostasis. And here's the dark part. Engaging in displacement aggression, physically lashing out, it actually lowers the perpetrator's glucocorticoid levels.
Are you serious? Lashing out biochemically calms them down.
Yes. The physical exertion, combined with the psychological perception of regaining control by dominating someone else, it acts as a biological release valve. You are physically offloading your physiological stress onto another living being. Literally, giving someone else an ulcer lowers the chances of you getting one yourself.
That is just incredibly bleak. We are biologically wired to feel better by making someone else feel worse.
It's a harsh reality of our neurochemistry.
But, it highlights how easily we understand hot-blooded actions. We can wrap our heads around a crime of passion or lashing out because you're stressed at work. It's relatable, even if it's morally wrong. But let's flip the script for a second. What about acts of goodness? Why does cold-blooded goodness unnerve us so much?
That is a profound question Sapolsky explores, and it cuts right to the core of human expectation. We expect our most laudable acts to be warm-hearted, deeply empathetic, and filled with positive emotion. So when we encounter detached, affectless altruism, it feels unnerving.
It almost feels like an oxymoron. Cold altruism.
Precisely. The book references a subset of people known as extreme altruists. These are Samaritans who voluntarily donate their kidneys, not to a dying child or a beloved spouse, but to complete strangers.
And people often react to these donors with deep suspicion, right? Not awe.
Yes, deep suspicion. People wonder if the donor is secretly being paid under the table, or if they have a pathological need for attention, or if they're just mentally unstable. The sheer dispassionate calculation of giving away an organ to a stranger makes us profoundly uncomfortable.
It just doesn't fit our narrative of what a hero looks like.
Sapolsky also recounts this experience at a conference with highly experienced Buddhist monk meditators. A scientist asked a monk if he ever stops meditating simply because his knees hurt from sitting cross-legged for so long.
And what did the monk say?
The monk replied that he might stop sooner than planned, not because he is overwhelmed by the pain, but simply as a dispassionate act of kindness to his knees. It is a cool, commendable, but completely alien way of processing the world.
It really is from another planet. We desperately want our heroes to be passionate, just like we expect villains to be passionate. It reminds me of that haunting insight from Elie Wiesel, the Holocaust survivor.
Oh yes, that's a powerful quote.
He said that the opposite of love is not hate, its opposite is indifference. And biologically, the neurobiology of strong love and strong hate actually share a lot of architecture, right? There are both highly aroused, high-stakes physiological states.
They absolutely are. It's that cold, detached state, whether it results in calculating violence or extreme kidney-donating altruism, that truly sits apart from the rest of human experience.
And to truly understand the biological machinery behind both those passionate reactions and the detached calculations, we have to move our timeline. We have to look at what happens precisely 1 second before the behavior occurs.
Which brings us to Chapter 2, 1 second before. This is the realm of the brain. The brain is the final common pathway. Whatever happened a million years ago or an hour ago, it ultimately has to funnel through the physical neurobiology in that final second to command the muscles to move.
And to navigate this absolute labyrinth of neural pathways, we need a map. Sapolsky introduces this highly influential model from the 1960s by Paul MacLean called the triune brain model.
Now, we should add a caveat that modern neuroscience views this as an oversimplification. But functionally, it gives us 3 incredibly useful domains to navigate. Think of it as layer 1, layer 2, and layer 3.
Okay, let's break down the architecture. Layer 1 is the evolutionarily ancient part of the brain, right? Located at the base where the spinal cord enters the skull.
Exactly. You find this core architecture in species ranging from geckos to human beings. This layer is entirely about automatic regulatory functions. It keeps you alive without you ever consciously thinking about it.
Like a biological autopilot.
Spot on. If your core body temperature drops, neurons in this region sense the change and automatically command your muscles to shiver to generate heat. If your blood glucose plummets, this region generates a profound feeling of hunger. It is the deep reptilian core of survival.
Then we step up to layer 2, which MacLean conceptualized as a more recently evolved region that expanded with mammals. This is the limbic system, and its primary currency is emotion.
Right. If you see something terrifying, layer 2 reacts and sends commands down to ancient layer 1, making you shiver, not from a drop in temperature, but from fear. If a rodent smells a predator, like a cat, neurons in layer 2 hit the panic button and force layer 1 to initiate a stress response.
And finally, encompassing all of this, we have layer 3, the neocortex, sitting on the upper surface of the brain like a heavily folded helmet.
And proportionally, primates, specifically humans, devote massive amounts of metabolic energy to this outer layer. This is the home of cognition, memory storage, abstract thought, and philosophy.
It's the layer that allows you to read a scary passage in a Stephen King novel, which, let's be real, is just processing abstract ink symbols on paper. But layer 3 signals layer 2 to actually feel frightened, which prompts layer 1 to make your heart race.
Exactly. It lets you watch a movie and feel genuine sadness for fictional characters, even though layer 3 simultaneously knows they're just pixels on a screen.
Wait, I have to push back on the metaphor people usually use for this, because people often describe the triune brain like a perfectly layered cake or an archaeological dig where the old stuff is buried and untouched by the new stuff. But evolution doesn't just stack new brain layers on top without touching the old ones, right?
You are entirely correct to push back. The layered cake metaphor is disastrously misleading. The layers are intricately intertwined, and the flow of information is not just top-down from cognition to emotion. The flow is highly bidirectional.
The physical pathways loop back on themselves.
Constantly. Let's think about a fascinating example. Imagine you are holding a warm cup of coffee. You're introduced to a total stranger, and later you're asked to rate their personality.
Okay.
Because your hands were physically warm, you are statistically far more likely to judge that stranger as having a warmer, kinder personality. Your physical temperature just hijacked your moral judgment.
That is just mind-bending. Your physical sensation of temperature is hijacking your social morality. How does that physically happen in the brain?
Let's look at the mechanism. Temperature is a basic, vital input processed down in the core of layer 1 in a region called the insula. But that ancient thermal sensor is physically hardwired into the advanced layer 3 cognitive circuitry that evaluates complex social judgments.
So the brain is efficiently using the same physical pathway that processes literal thermal cold to process metaphorical social coldness.
Exactly. Your ancient biological sensors are directly dictating your advanced philosophical assessments. Thought, emotion, and automatic bodily functions are completely inseparable.
So to really understand how these intertwined layers drive our best and worst behaviors, we need to look much closer at the bridge between them. We need to look at layer 2, the limbic system, the emotional heart of the brain.
And, the history of how we even discovered the limbic system's function is this fascinating story of scientific tunnel vision. In the early 20th century, neuroscientists examining the brains of rats noticed 2 gigantic prominent lobes at the front, the olfactory bulbs, which process smell. They traced the neural connections from these bulbs and found they led straight into the structures of layer 2. So, operating in a purely anatomical bucket, they named this entire region the rhinencephalon, which translates to the nose brain.
The nose brain. They thought its only job was smelling.
They concluded its sole, dedicated purpose was processing odors. Meanwhile, other researchers in the 1930s and 40s, scientists like MacLean and Papez, were studying primate behavior. They discovered that if you surgically lesioned these exact same structures, it caused massive abnormalities in social behavior and aggression.
So they called it the limbic system and declared it the center of emotion. You have 2 camps looking at the exact same brain region. One says it's for smelling, the other says it's for feeling.
Yes, until someone finally stepped back and pointed out the blindingly obvious biological context. For a rat, emotion and olfaction are nearly synonymous.
Oh, wow, because they experience the world through their noses.
Right. Almost every single environmental stimulus that elicits an emotional response in a rodent, whether it's the paralyzing fear of a predator, sexual arousal, or territorial aggression, it comes entirely through its nose. In primates, our limbic system is far more informed by our eyes, but the neural architecture remains identical.
So there isn't like a singular cluster of cells that just makes you feel nostalgic and another that makes you feel jealous?
No, these regions form immensely complex, intersecting circuits of electrical excitation and chemical inhibition. And the one thing all these diverse limbic structures share, the central organizing principle of layer 2, is a desperate desire to influence one specific structure, the hypothalamus.
Let's dig into that. Out of all the complex real estate in the brain, why is that tiny structure, the hypothalamus, the absolute VIP?
Because the hypothalamus is the physical bridge between the emotional chaos of layer 2 and the core regulatory machinery of layer 1. The hypothalamus sends immense neural projections down into the ancient midbrain and brainstem. These primitive regions collectively form the autonomic nervous system, or the ANS.
And when the limbic system experiences terror, it doesn't just sit there feeling scared. It uses the hypothalamus as a megaphone to scream at the midbrain, demanding it alter the physical state of the entire body.
Exactly. People usually describe the autonomic nervous system using the analogy of a gas pedal and a brake pedal. But I prefer to think of it as a city's emergency power grid.
Oh, I like that. Violently redirecting resources based on the environment.
Yes. The ANS has two main divisions. First, you have the sympathetic nervous system, or the SNS. This is your emergency power grid diversion. It mediates the body's response to highly arousing critical circumstances. Medical students learn the SNS mediates the 4 Fs: fear, fight, flight, and sex.
So when the SNS detects a crisis, it triggers neurons that project down the spine. In a fraction of a second, these nerve endings release norepinephrine, right?
Correct. And simultaneously, it signals the adrenal glands to dump epinephrine, adrenaline, into your bloodstream. Heart rate skyrockets, blood pressure surges, and glucose is violently diverted away from long-term projects and pumped directly into your skeletal muscles so you can run or fight.
And what about the other side of the grid, the infrastructure maintenance side?
That is the parasympathetic nervous system, or the PNS. This system projects from entirely different midbrain nuclei, primarily through the vagus nerve, and it uses a different neurotransmitter, acetylcholine. The PNS is all about calm vegetative states.
So it slows the heart down and promotes digestion.
Right. If you are running from a lion, your SNS actively shuts down your digestion because you don't need to waste metabolic energy processing breakfast if you're about to become lunch. But once you are safe, the PNS kicks back in to restore balance.
And this has profound moral implications, doesn't it? If you encounter someone in terrible pain and your SNS emergency grid activates, your own physical distress will likely trigger a flight response, preventing you from helping. But if your PNS infrastructure grid engages, keeping your heart rate steady, you are biologically far more capable of stepping in with compassion.
Exactly. This architecture sets the stage perfectly. We have the autonomic core managing the power grid, the emotional limbic system processing the environment, and the cognitive cortex trying to make sense of it all. And sitting directly inside that emotional limbic system is perhaps the most notorious structure in behavioral biology, the amygdala.
The hub of fear, vigilance, and violence.
The amygdala is the archetypal limbic structure. It consists of two almond-shaped clusters nestled deep under the cortex. When neuroscientists look for the biological root of our most violent behaviors, the evidence points overwhelmingly to the amygdala.
Because if you insert recording electrodes into an animal's amygdala, the neurons fire wildly precisely when the animal is being aggressive.
Right. And in humans, viewing anger-provoking pictures causes the amygdala to metabolically light up. But moving beyond mere correlation to causation, if you surgically lesion the amygdala in an aggressive animal, its aggression plummets to near zero. Conversely, if you stimulate those neurons with a tiny electrical current, it triggers instant unprovoked rage.
And we don't just see this in animal models. There are incredibly dark, real-world human case studies that bear this out with chilling clarity. Charles Whitman is a prime example.
Yes, the Texas Tower sniper in 1966.
He murdered his wife and mother, then barricaded himself in a clock tower and shot dozens of innocent people. But when you look at his background, the neurobiology becomes central to the story. He wasn't a lifelong criminal. He was an Eagle Scout, a former Marine, an engineering major with a genius IQ.
But in the year leading up to the massacre, he sought medical help, complaining to doctors about agonizing headaches and violent, uncontrollable impulses that were completely out of character.
In his suicide note, he literally begged for an autopsy of his brain to find out what was physically wrong with him. And that autopsy revealed a glioblastoma, a highly aggressive brain tumor pressing directly on his amygdala.
Now, Sapolsky is extremely careful to point out that we cannot reduce human tragedy to a simplistic formula of amygdaloid tumor equals mass murderer. The biology is deeply contextual. Whitman also grew up being violently abused by his father, representing a volatile mix of severe neurological impairment and deep childhood trauma.
But the physical structural pressure on the exact neural hub of aggression was undeniable. The text also details the case of Ulrich Meinhof, right?
Yes, a key founder of the Red Army faction, a violent terrorist group in the 1970s. She was a journalist, but became increasingly radicalized and violent. After she died, an autopsy revealed that remnants of a supposedly benign brain tumor she had removed years earlier, along with the surgical scar tissue, were impinging directly on her amygdala.
It is deeply unsettling to think about how vulnerable our entire moral identity is to a few millimeters of tissue pressure. But here's the major plot twist regarding the amygdala. If you ask an expert what behavior the amygdala is known for, they wouldn't say aggression.
They would say fear. That is the most profound, counterintuitive paradox of the limbic system. The brain region most causally involved in generating violent aggression is fundamentally the exact same region most involved in feeling terrified and anxious.
The evidence is identical, right? Lesion it, fear vanishes. Stimulate it, panic ensues.
Exactly. The human amygdala preferentially responds to fear-evoking stimuli, even if those stimuli are flashed on a screen in mere milliseconds, completely below our conscious detection. In individuals with PTSD, the amygdala is chronically overreactive, it's incredibly slow to calm down, and it actually physically expands in size over time due to the sustained barrage of trauma.
I want to really dig into this because the amygdala doesn't just react to innate evolutionary fears. Like a rat born in a lab that has never seen a cat will instinctively freeze if you waft cat pheromones into its cage. That innate fear is managed by the central core of the amygdala. But the amygdala also learns fear.
Yes, and that mechanism of learning occurs in a surrounding region called the basolateral amygdala, or the BLA. The BLA operates primarily through Pavlovian fear conditioning.
Okay, let's trace that pathway.
If you expose a rat to a painful electrical shock, the central amygdala activates and the rat freezes. But if you play a completely neutral tone right before the shock, over and over, the sensory inputs for the tone and the pain wire together in the BLA.
The BLA physically alters its synapses.
Exactly. Eventually, just playing the tone alone causes the BLA to signal the central amygdala to panic. The BLA learns fear. And crucially, it struggles with fear extinction, the active process of unlearning that the tone means danger once the shocks stop.
So if we look at this practically, think of the amygdala like a highly skeptical, overly aggressive security guard. Our default biological state as human beings is actually to trust one another.
Right, baseline trust.
The amygdala is the security guard whose entire job is to be actively trained by negative experiences via the BLA to learn vigilance and distrust. Without a functioning BLA, we would default to pathologically trusting everyone, even people who repeatedly exploit us.
And the reason that security guard is uniquely positioned to overreact and trigger violence is because of a very specific evolutionary quirk in how it receives information.
The sensory express lane?
Yes. Typically, sensory information, like seeing a shape in the dark, enters your eyes, goes to the thalamus, the brain's relay station, and then goes all the way to the visual cortex at the back of your head. The visual cortex slowly processes the image, layer by layer.
Decoding the lines, the shadows, figuring out what it's looking at. And once it has a clear picture, it sends a message to the amygdala.
Which is highly accurate, but it takes precious milliseconds. However, evolution built a biological shortcut. A small bundle of neurons routes some sensory information directly from the thalamus, completely bypassing the cortex, and sends it straight into the amygdala.
It doesn't wait for the high-resolution picture.
Exactly. The amygdala gets informed about a potential threat before your conscious cognitive cortex even has a clue what's going on. The evolutionary trade-off is extreme. You gain life-saving speed, but you sacrifice immense accuracy. The thalamus just sends a blurry, low-resolution shape.
And this shortcut is the exact tragic mechanism that causes the brain to perceive a harmless, dark-colored cell phone in someone's hand as a deadly handgun in a split second.
Yes, the amygdala receives the blurry input, screams gun, and commands the autonomic nervous system to react with defensive aggression before the visual cortex can finish rendering the image and say, wait, stand down, it's just a phone.
That split-second biological glitch is responsible for so much profound tragedy. It highlights that fear and aggression are intertwined. We strike out because we are terrified. But Sapolsky points out a massive exception to this rule: the violent psychopath.
And understanding psychopaths shatters the idea that all violence comes from fear. While fear increases aggression in most individuals, violent psychopaths are the complete physiological antithesis of fearful. When exposed to fear-evoking stimuli, they show almost zero autonomic arousal.
Their heart rates don't spike, they don't sweat.
Nothing. And when we look at their brain architecture, their amygdalae are significantly smaller than normal and show deeply muted metabolic responses. This proves that not all aggression is rooted in the amygdala's panic response. Psychopathic violence is purely instrumental.
It is cold-blooded, emotionless, and remorseless. They lack the physiological arousal of fear or the hot-blooded rush of anger. Which brings us to the final major brain region we need to explore in this first part of our deep dive. We've looked at layer 1 and 2, now we move up to layer 3, to the newest machinery of human cognition, the frontal cortex.
And specifically the prefrontal cortex, or the PFC. If the amygdala is the panicking security guard, the frontal cortex is the
The frontal cortex is fundamentally distinct, right? Most notably, it's the absolute last part of the brain to fully mature. It doesn't finish its synaptic pruning until we are in our mid-20s.
That's right. Its core defining function is deceptively simple to say, but incredibly hard to execute, making you do the harder thing when it is the right thing to do. It is the undisputed center of executive function. It manages working memory, keeping complex rules online in your head.
And crucially, it handles impulse control, sending inhibitory signals down to actively suppress the reflexive urges bubbling up from the amygdala. When you want to blurt out a rude thought but you bite your tongue, that is your frontal cortex metabolically burning glucose to do the harder thing.
It's the ultimate brake pedal, and we know exactly what happens when those brakes are physically removed. The text touches on the famous neurological case of Phineas Gage,
the railroad construction foreman in the 1840s. He was known for being polite and responsible. Then an explosion drove an iron tamping rod straight through his cheek and out the top of his skull, physically obliterating a massive portion of his frontal cortex.
Miraculously, he survived. He could walk and talk, but his identity was irreversibly changed. Without his frontal cortex to exert top-down impulse control, he became profane, erratic, and utterly incapable of planning for the future. He could no longer do the harder thing.
The emotional impulses of his limbic system were running the show with zero oversight. But Sapolsky makes a nuanced distinction here. The frontal cortex is not just one monolithic, rational calculator. It has highly specialized subdivisions. We must contrast the dorsolateral prefrontal cortex, the DLPFC, with the ventromedial prefrontal cortex, the VMPFC.
Let's look at the DLPFC first. The dorsolateral prefrontal cortex is the cold, rational, strictly cognitive center. It solves complex math equations, organizes your calendar, and analyzes pure utilitarian outcomes. The VMPFC, however, sits physically right at the intersection between the cognitive frontal cortex and the emotional limbic system.
It is the emotional interface. It takes the cold data from the DLPFC and marries it to the hot data coming up from the amygdala.
Exactly. And we can see this battle perfectly illustrated in the runaway trolley problem. Imagine a trolley is hurtling toward 5 workers who will be killed. You can pull a lever to divert it onto a sidetrack, killing only 1 worker. Do you pull the lever?
Between 70 and 90% of people say yes. They do the math. 5 lives saved minus 1 life lost equals a net positive. In the brain scanner, making this decision heavily activates the cognitive utilitarian DLPFC. It's a math problem.
But then the scenario changes. Same trolley, same 5 workers, but no sidetrack. You were standing on a footbridge right over the tracks next to a large man. The only way to stop the trolley is to physically push the man off the bridge. His mass will stop the trolley, saving the 5, but he will die.
From a purely mathematical standpoint, the equation is exactly the same. 1 life traded for 5. But how do human beings actually react?
Utterly differently. Between 70 and 90% of people say absolutely not. They refuse to push the man. And when we look at their brains in an fMRI, the physical contemplation of pushing a human being with your own hands causes the emotional VMPFC and the amygdala to scream in protest.
Ultimately, the intuitive emotional regions completely veto the cold math of the DLPFC. The ends no longer justify the means when the physical action feels so biologically wrong.
And this ties directly into Antonio Damasio's somatic marker hypothesis. Damasio proposes that when we face a complex moral decision, the limbic system rapidly runs an emotional simulation of what the future outcome would literally feel like.
And it reports that gut feeling back to the VMPFC via pathways like the vagus nerve. If you contemplate pushing the man, your limbic system simulates the horror, your sympathetic nervous system gives you a clammy sweat, your stomach drops, and your VMPFC registers that as a giant red flag.
That physiological gut feeling allows us to navigate moral landscapes. Without the VMPFC to read those bodily emotional markers, a person becomes a hyper-rational sociopath, perfectly willing to push anyone off a bridge if the math checks out.
The collaboration between the emotional limbic system and the cognitive frontal cortex is a delicate dance, but it's also terrifyingly fragile. Think about a day where you've been in stressful meetings, skipped lunch, and you snap at your partner over something tiny. That is your frontal cortex literally running out of metabolic energy, suffering from cognitive load, and failing to suppress the amygdala.
It's all biological mechanics.
We've spent this time mapping the split-second machinery that dictates our actions. We've seen how pulling a trigger or extending a hand might be dictated by the physical size of an amygdala, a high-speed sensory shortcut, or simply the cognitive load draining the prefrontal cortex in that exact millisecond.
It really reframes everything.
It does, and that leaves us with a deeply unsettling question to ponder. If we are simply the sum of this biological machinery, if our most profound moral choices are just neurochemical dominoes falling 1 second before the act, where exactly does the self reside? If a few millimeters of tumor tissue, or a rapid drop in blood glucose, or a sensory glitch can turn a choir boy into a killer, how do we fairly judge the 1 second before of a crime? As we leave you today, ask yourself, are your morals a product of your unshakable character, or are they just a byproduct of a well-fed, highly functioning frontal cortex that hasn't been tested yet?
This brings us to the end of part 1. In the upcoming part 2, we will expand our biological lens further back in time. We'll explore how real-time sensory triggers, hormonal rushes hours to days prior, early childhood development, genetics, and culture shape the brain's split-second decisions. Stay tuned for the next part of this deep journey. Imagine you're a juror in a murder trial. The evidence has been meticulously presented, the closing arguments have been made, and you're just sitting there in the deliberation room.
Right, you're weighing everything logically.
Exactly. You feel completely confident in your objective, rational assessment of the defendant's guilt. You weighed the facts and you made a logical choice. But what if your moral judgment was actually being dictated by how uncomfortable the chair you're sitting in happens to be?
Or, the ambient smell of the room?
Yeah, or maybe the specific, colorful adjectives the prosecutor used 20 minutes ago. What if all of that secretly rewired your brain's threat response before you even consciously formed a thought?
It's a really unsettling premise, but that's exactly what the biology shows us.
Welcome back to Fact Horizon. In part 1 of our breakdown of Robert M. Sapolsky's Behave, the biology of humans at our best and worst, we examined what happens in the nervous system 1 second before a behavior occurs, focusing on the amygdala and the prefrontal cortex.
Which really set the foundation for understanding our immediate reactions.
Right. This is part 2 of our deep dive into Behave. In this episode, we move further back along the timeline of causality, examining how short-term environmental triggers, endocrine signaling, and neuroplasticity condition our behavior. We will focus on seconds to minutes before, sensory triggers and unconscious cues, hours to days before, hormones and endocrine influences, and the neurobiology of moral choice, us versus them, and empathy.
And understanding this expanded timeline is really where the illusion of complete, unvarnished free will begins to fracture. It's where the true complexity of human agency comes into focus.
Because, if you're joining us for this continuation, it's because you recognize that human behavior cannot just be isolated to a single moment of conscious decision-making.
Absolutely. You understand that the invisible forces shaping your actions, like the biological and environmental variables operating beneath the threshold of your awareness, are what truly govern the human experience.
So let's trace this back. In part 1, we established the absolute final mechanism, right? The split-second firing of neurons, with the prefrontal cortex desperately trying to provide top-down regulation to a highly reactive amygdala.
Right, the brakes trying to stop the engine.
Exactly. But a neuron doesn't just fire spontaneously in a vacuum. It reacts to a stimulus. So we need to step back into the seconds to minutes before a behavior occurs and enter the subliminal world of sensory triggers.
And the architecture of how we process these triggers reveals a critical evolutionary compromise, the compromise between speed and accuracy.
Speed versus accuracy. Break that down for us.
Well, to understand how vulnerable we are to our environment in the seconds before we act, we really have to look at the neuroanatomy of sensory processing. So let's trace a visual stimulus.
Okay, like seeing something in front of you.
Right. Normally, light hits your retina, travels down the optic nerve, and arrives at the thalamus. The thalamus is, it's basically the brain's central relay station. It takes that raw sensory data and routes it to the primary visual cortex at the back of your brain.
And the visual cortex is where the heavy analytical lifting happens.
Right.
It processes the lines, the contrast, the depth, and then it passes the image through subsequent cortical layers to actually recognize shapes, faces, and context.
Exactly. It's a Brilliant, highly sophisticated rendering process, but here's the catch. It is relatively slow.
Too slow for survival sometimes.
Yeah, exactly. By the time the visual cortex has assembled the pixels, cross-referenced them with your memory, and sent a message down to the amygdala saying, hey, that object is a threat, precious milliseconds have passed. And in a hostile Pleistocene environment, those milliseconds are literally the difference between life and death.
So evolution built a shortcut.
A direct wire, a neural projection that goes from the thalamus straight to the basolateral amygdala, completely bypassing the visual cortex.
Bypassing the analytical part entirely.
Yes. A portion of the sensory data diverges at the relay station and takes this express route. And the synapses in this pathway are highly excitable. They're heavily myelinated for fast transmission, and they're tuned for one specific purpose, which is threat detection.
Wow.
It allows the amygdala to initiate a massive sympathetic nervous system response before the frontal cortex even knows what it is looking at.
Which, I mean, that makes perfect sense if you're a hominid on the savannah. If you see a long, thin, curved shape in the grass out of the corner of your eye, you really don't want your visual cortex taking its time to determine the exact species of venomous snake, or verifying if it's actually just a harmless stick.
Right, because jumping back from a stick just costs you a few calories. But failing to jump back from a snake costs you your life.
But when you place that exact same biological hardware into the complex, ambiguous modern human world, the results can be just devastating.
They really can, because this thalamic shortcut is extraordinarily fast, but it is also highly inaccurate. The amygdala receives low-resolution, incomplete data, and it just acts on it with absolute certainty.
Which explains so many tragic errors.
Yes. This is the biological mechanism behind some of the most tragic errors in judgment in society. It's the neurobiology of, say, a police officer operating under high stress with a highly aroused amygdala, misidentifying a cell phone or a wallet being pulled from a pocket as a handgun.
The shortcut triggers the motor outputs to fire. The behavior occurs.
Exactly. And then, a fraction of a second later, the visual cortex finally finishes rendering the high-resolution image, sending that agonizing realization that it was just a phone.
God, it's just chilling to realize how captive we are to our own sensory routing. And this immediate environment isn't just visual, is it? One of the most fascinating layers Sapolsky explores in the source material is olfaction. Scent.
Scent is huge. We tend to think of humans as mostly visual and auditory creatures, largely divorced from the scent-driven worlds of dogs or rodents.
Right, because our noses aren't as good.
Well, it's a common misconception, primarily because our olfactory bulb is proportionally smaller. To give you some anatomical context, roughly 40% of a rat's brain is devoted to processing smells.
40%. That's almost half the brain.
Yeah, and in humans, it's about 3%. Early neuroanatomists actually referred to the emotional center of a rodent's brain as the rhinencephalon, which literally translates to the nose brain. For a rat, emotion and olfaction are neurobiologically inseparable.
But just because our hardware is smaller doesn't mean it isn't potent. I mean, that 3% still projects heavily and directly into the limbic system, doesn't it?
It absolutely does, and it operates almost entirely on a subliminal level. We are constantly absorbing and reacting to chemical signals in the air without any conscious awareness at all.
And the most profound demonstration of this in the literature has to be the tandem skydiving sweat study. I want to spend some real time on the methodology of this study because it just perfectly illustrates how invisible cues hijack the brain.
It's an incredible study. The researchers wanted to isolate the specific chemical signature of human terror. So they gathered sweat from volunteers under two strictly controlled conditions.
Okay.
In the first condition, volunteers ran on a treadmill. This produced just normal exercise-induced sweat. But in the second condition, they collected sweat from volunteers who were doing their very first tandem skydive.
Which is such a crucial distinction, because in a tandem skydive, the novice is strapped to an instructor. The instructor does all the physical exertion, pulling the cords and steering.
Right. The novice is just along for the ride.
So the sweat they produce isn't from physical effort.
Yeah.
It is pure, unadulterated fear. It's a completely different endocrine response.
Exactly. The researchers then took these two sets of swabs, the exercise sweat and the terrified sweat, and placed test subjects in an fMRI scanner. The subjects were asked to sniff the swabs.
And what did they consciously smell?
Well, if you asked them consciously, they could not articulate any difference between the two smells. To their conscious mind, it was just faint body odor.
But the fMRI scans told a completely different story, didn't they?
The neural divergence was massive. When the subjects inhaled the terrified sweat, their amygdala immediately lit up. Furthermore, the researchers measured their startle reflexes, like, the involuntary eye blank response to a sudden noise, and found it was significantly exaggerated.
So their nervous systems were mobilizing for danger just from a smell?
Exactly. But the most unsettling part of the study is how it actually changed their perception of other people.
Tell us about the faces.
While in the scanner, the subjects were shown images of human faces that had been digitally morphed to display ambiguous expressions, so somewhere between neutral and angry.
Okay.
The subjects who had inhaled the terrified sweat were statistically much more likely to interpret those ambiguous neutral faces as hostile and threatening.
Wow. It's like having a hyperactive smoke detector that goes off because of a dusty breeze, but it convinces you the house is on fire.
That's a perfect analogy.
I mean, if your brain detects the chemical signature of fear from someone else in the room, it bypasses your logic entirely and concludes, well, if they are terrified, there must be a predator. Therefore, I need to be hypervigilant.
Right, and suddenly you're interpreting a completely neutral glance from a coworker or a stranger as a deliberate threat. You become prone to preemptive aggression, all because of an invisible molecule you didn't even know you inhaled.
That makes me push back a bit on the concept of free will here. I mean, if we're aggressively reacting to invisible smells and split-second visual errors, are we just biological automatons in these moments?
It certainly challenges the idea of absolute free will, and, that environmental priming extends beyond what we see and smell. The brain is also constantly monitoring the internal environment of the body.
Right, through interoception.
Yes, the process of interoception. Data about your heart rate, your body temperature, your gastric motility, it all streams into the brain, specifically into regions like the insular cortex and the anterior cingulate cortex.
And those are the exact same regions involved in processing social and emotional information.
This overlap is absolutely vital. Interoceptive sensations act as a powerful context for behavior. Consider physical pain, for instance. Pain does not independently generate aggression. If you take a generally passive individual and give them a sharp gastric pain, they don't suddenly become a violent criminal.
Right, pain isn't just a light switch that turns on violence.
But it operates as a profound amplifier. Pain lowers the threshold for aggressive circuitry to fire. If an individual already has a predisposition toward reactivity, introducing an uncomfortable stimulus like an achy tooth, a painfully cold room, or a highly uncomfortable chair magnifies that pre-existing tendency.
So the discomfort bleeds over.
Exactly. When you sit in an uncomfortable chair, the nociceptors sending pain signals to your brain share neural real estate with the circuits evaluating moral judgments. Consequently, subjects in these conditions routinely judge the transgressions of others far more harshly. Their physical discomfort seamlessly becomes their social evaluation.
The metaphorical line between physical discomfort and moral rigidity practically vanishes. And the sources point out that this sensory priming isn't even limited to physical sensations. The language we are exposed to seconds before a decision acts as an auditory prime that rewires our moral calculus. Oh, the neurobiology of semantic priming is incredibly robust. Trial lawyers have exploited this intuitively for centuries, but modern neuroimaging shows us exactly how it works.
What happens in the scanner?
Well, if a prosecutor describes a defendant's action using words like blameworthy or forbidden, it activates certain cortical networks associated with rule following. But if they use a word like wrong or disgusting, it engages the anterior cingulate cortex and the insula much more aggressively.
The language itself acts as a sensory trigger. The jury hears the word disgusting, and their brain biologically prepares to expel a pathogen, resulting in a much harsher punitive judgment.
Exactly. The environment in the seconds to minutes before behavior is just an invisible minefield of visual shortcuts, subliminal odors, internal aches, and weaponized adjectives. So if these immediate sensory triggers act as the software executables that initiate our behaviors, what dictates the state of the operating system they are running on? What chemically primes the brain's hardware to be highly sensitive to those triggers in the first place?
That brings us further back in the timeline, exploring the hours to days before a behavior. We enter the realm of endocrinology.
Hormones.
Yes. And to understand how hormones condition our reality, we really have to start by dismantling the most entrenched mythological construct in all of biology, the nature of testosterone.
Oh, the cultural narrative surrounding testosterone is so pervasive. It's viewed as this toxic, malevolent serum. We blame it for road rage, bar fights, warfare, and toxic masculinity. The assumption is that testosterone is basically the biological equivalent of aggression.
It's a very convenient myth.
Uh-huh.
But endocrinologists view causality through a much more rigorous lens, primarily using subtraction and replacement experiments.
Let's break down the mechanics of those experiments because they reveal exactly how the testosterone causes violence. Myth totally falls apart.
Right. If you want to prove that a hormone from the testes directly causes aggression, you first perform a subtraction. You castrate a male animal, removing the source of the hormone.
And what happens?
When researchers do this across various mammalian species, average levels of aggression plummet. This confirms that testicular hormones are definitely involved in the behavior.
Okay, so that's step one.
The second step is replacement. You administer synthetic testosterone to the castrated animal, and pre-castration levels of aggression return.
Now, on the surface, that sounds like an open and shut case. Subtraction removes the behavior, replacement restores it. So testosterone causes it, right?
It does seem that way, until you look closer at the data from the subtraction phase. When you castrate the animal, aggression plummets, but it crucially does not drop to zero. A significant baseline of aggressive behavior remains completely independent of testosterone.
Wait, if the hormonal fuel is entirely gone, where is that remaining aggression coming from?
It comes from neuroplasticity. The remaining aggression correlates perfectly with the individual's prior social experience.
Yes. If a male monkey had extensive experience being highly aggressive and dominant before the castration, that aggressive behavior persists much longer after the castration. The behavior has shifted from being hormonally driven to being a structurally ingrained habit.
Wow, the brain physically changes.
Exactly. The neural pathways in the amygdala and the motor cortex have physically wired themselves to respond aggressively to certain social cues. The brain remembers how to be violent, even without the endocrine trigger. That is the first massive fracture in the myth. And the second fracture involves how the brain interprets normal fluctuations in the hormone, right? People assume that if person A has a higher baseline level of testosterone than person B, person A must be more aggressive.
And across humans, primates, birds, and fish, the data resoundingly rejects that assumption. Within the normal physiological range, the brain simply does not pay attention to baseline fluctuations in testosterone. Having slightly more testosterone than the guy sitting next to you does not predict that you will be more aggressive.
So, to use an analogy, testosterone doesn't write the music. It doesn't compose the violent song. It just acts as a volume knob, turning up whatever track is already playing in the brain's hardware.
That analogy perfectly captures what endocrinologists call the contingent and amplifying effects of the hormone. Testosterone does not invent new aggressive patterns. It exacerbates pre-existing tendencies.
Give us an example of how that amplifying effect works.
Imagine a stable dominance hierarchy of 5 male monkeys. Number 1 is the alpha, number 2 is the beta, all the way down to number 5, who is the lowest ranking male. Naturally, number 3 is submissive to 1 and 2, but he aggressively dominates 4 and 5.
Okay, the middle management monkey. Now, what happens if you take that middle ranking monkey, number 3, and artificially inject him with massive doses of testosterone? If the cultural myth were true, he should become an uncontrollable brute and challenge the alpha, right?
But he never challenges number 1 or number 2. The testosterone does not give him the audacity to upend the social order.
So what does he do?
Instead, he just becomes an absolute tyrant to number 4 and number 5. The hormone did not create a new behavioral trajectory. It simply lowered the threshold for the amygdala to fire in response to the social cues it was already primed to react to. It amplified the existing hierarchy.
If baseline levels don't predict aggression and massive injections don't create new aggression out of thin air, when does testosterone actually matter?
This is where we look at the brilliant framework proposed by behavioral endocrinologist John Wingfield, known as the challenge hypothesis.
The challenge hypothesis.
Right. Wingfield demonstrated that testosterone levels are not static. They rise dynamically at the exact time of a challenge to an individual's status or reproductive success.
That's it, situational.
Highly situational. If a dominance hierarchy becomes unstable, testosterone spikes. In humans, testosterone surges in anticipation of a competition, whether it's a wrestling match, a game of chess, or even just watching your favorite sports team play in a championship.
So it's deeply tethered to the psychology of status.
Precisely. And when testosterone rises in response to that challenge, it makes aggression more likely. But And here's the absolute crux of the entire paradigm. The challenge hypothesis dictates that rising testosterone prompts whatever behaviors are needed to maintain status.
Whatever behaviors are needed. In a traditional primate society, maintaining status usually requires physical violence, a terrifying stare, a physical assault. So we naturally associate the hormone with the violence. But humans construct highly complex social environments. What happens if you engineer a society where defending your status requires you to be exceptionally kind?
This leads us to one of the most paradigm-shifting experiments in behavioral economics. The ultimatum game study conducted by researchers in Zurich.
The methodology here is brilliant. Let's break down exactly how the ultimatum game forces the brain to calculate status.
So in the ultimatum game, two players interact. Player A, the proposer, is given a pot of money, say $100. Player A must propose a way to split that money with Player B, who is the responder. They can offer a fair 50-50 split, or they can be greedy and offer a 90-10 split.
But the catch is Player B's power. If Player B accepts the offer, both players get the money as proposed. But if Player B rejects the offer, the money is literally destroyed, and neither player gets a dime.
Right. Now, classical, purely rational economic theory states that Player B should accept any offer greater than zero. I mean, $1 is better than nothing, right?
But human psychology does not operate on pure economic rationality. We calculate fairness and social reputation. If Player A offers a humiliating 90-10 split, the insular cortex of Player B lights up with disgust, and they will spitefully reject the offer, willingly burning their own money just to punish the norm violator.
Exactly, because accepting a greedy offer feels like subordination. It damages your status.
So the proposer has to navigate that.
Yes. In the context of this game, Player A's status, reputation, and financial success rest entirely on being perceived as fair and generous. The dorsolateral prefrontal cortex is constantly calculating the optimal offer to maintain social standing while maximizing reward.
And this is where the hormones come in.
Right. The Zurich researchers took a group of proposers and administered synthetic testosterone to them before the game.
If the old cultural myth were true, those testosterone-fueled players should become hyper-aggressive, greedy, and demand the lion's share of the cash.
But the exact opposite occurred. The subjects who received testosterone made significantly more generous and fair offers than the control group receiving a placebo.
They fiercely competed to be the most pro-social.
Because in that specific, engineered microsociety, fairness was the currency of status. The testosterone did not make them violent. It made them hypervigilant about their social standing. It motivated them to execute whatever behavior was necessary to secure their reputation.
Wow.
The profound takeaway here is that if our world is plagued by male violence, the neurobiological root cause is not the existence of testosterone. The root cause is the frequency with which our cultures, our schools, and our institutions actively reward aggression with status.
That completely inverts the narrative. If we structured society to award the highest social prestige to acts of profound compassion, testosterone would actually be the most powerful pro-social hormone in human biology.
Absolutely. We blame our biology for the flaws in our cultural design.
Now, if testosterone is unfairly demonized, let's look at the hormone that receives universal, uncritical praise. Let's look at oxytocin. I playfully call this the groovy neuropeptide myth because the media just loves oxytocin. It is constantly branded as the cuddle chemical, the love hormone, the biological panacea for global conflict.
The groovy neuropeptide myth is just as neurobiologically incomplete as the toxic testosterone myth. It is true that oxytocin and its closely related non-peptide vasopressin are central to some of the most beautiful aspects of mammalian behavior.
Like mother-infant bonding.
Yes, they facilitate mother-infant bonding. They are the neurochemical foundation of monogamous pair bonding in certain species of voles. In humans, administering oxytocin can lower anxiety, increase the duration of eye contact, and enhance a person's ability to accurately read subtle emotional expressions.
It creates this beautiful, positive feedback loop. You experience a warm social interaction, your brain releases oxytocin, which lowers your amygdala's threat response, making you more trusting and pro-social, which leads to more warm interactions.
It acts as an amplifier of social competence and pro-sociality. But, much like testosterone only amplifying pre-existing aggression, oxytocin primarily amplifies pre-existing generosity. If you administer oxytocin to someone who is naturally selfish, it does not magically transform them into a philanthropist.
But there's a darker side too, right? The true dark side of oxytocin lies in its profound contingencies regarding social categorization.
The interaction between oxytocin and the us versus them dynamic is striking. Oxytocin makes us incredibly trusting, empathic, and cooperative, but only toward our perceived in-group. It binds us together.
And what happens with the outgroup?
When an individual under the influence of oxytocin interacts with an outgroup member, them, the hormone does the exact opposite. It actually fuels xenophobia.
The research by Karsten de Drew perfectly exposes this. Let's unpack his prisoner's dilemma study, because it demonstrates how oxytocin actually weaponizes loyalty.
De Drew, an incredible researcher in Amsterdam, set up a multi-round game of the prisoner's dilemma. In this game theory classic, you can choose to cooperate with the other player for a modest mutual benefit, or you can betray them for a massive personal gain, leaving them with nothing.
And he added a group dynamic.
Yes, he divided subjects into clear in-groups and out-groups, essentially creating competing teams.
When the subjects were given oxytocin, what happened to their strategy?
They became fiercely loyal to their in-group, just as the myth would predict. But they demonstrated that loyalty not by being universally peaceful, but by engaging in preemptive, aggressive backstabbing against the out-group.
So they attacked the other team.
The oxytocin heightened their paranoia toward them. They felt a profound need to protect their team, and the hormone drove the neurobiological calculation that the best defense was a devastating first strike. Oxytocin promotes what we call parochial altruism.
The bonding of the in-group fundamentally requires the vilification of the out-group. And de Drew followed this up with an even more unsettling study using the classic trolley problem.
The study isolates the ethnocentric effects of the hormone perfectly. The trolley problem is the famous moral dilemma, right? A runaway trolley is hurtling down a track toward 5 innocent people. The only way to save them is to physically push one heavy bystander onto the tracks, sacrificing the one to save the 5.
It forces the brain's emotional aversion to killing to battle against the prefrontal cortex's utilitarian math. De Drew gave this scenario to native Dutch students, didn't he?
He did. The 5 people tied to the tracks were left anonymous, but the one bystander who could be sacrificed was given a specific name. Half the time, the bystander was given a stereotypically native Dutch name, an in-group identifier like Dirk or Peter.
Oh.
The other half of the time, the bystander was given a stereotypically German name, like Helmut, or a Middle Eastern name, like Ahmed. Clear outgroup identifiers for this specific demographic.
Setting up a stark biological test of who counts as a human worth saving.
Exactly. When the Dutch students were given a placebo, their moral calculations were relatively consistent. But when they were administered oxytocin via a nasal spray, their ethnocentrism skyrocketed. The hormone made them significantly less likely to push Dirk or Peter onto the tracks. They fiercely protected their in-group.
But for Helmut or Ahmed?
They were perfectly willing, even eager to sacrifice Helmut or Ahmed.
But oxytocin isn't a universal love hormone. It's an agent of tribalism. From an evolutionary perspective, it evolved to help mammals identify their kin and fiercely protect them from competitors. It strengthens the walls of the tribe.
Which brings us to the third major endocrine player operating in the hours and days before behavior: glucocorticoids, stress hormones.
And stress, neurobiologically speaking, is an inverted U curve, meaning that a lack of stress isn't actually the goal.
Exactly. On the far left of the inverted U curve, you have zero stress. You are understimulated, bored, and lethargic. As you move to the peak of the curve, you encounter acute short-term stress. This is optimal stimulation.
That's the fight or flight we always hear about.
Right. This is the physiological state of sprinting away from a predator, or the intense focus required to play a competitive sport. Your adrenal glands pump out epinephrine and glucocorticoids. Your heart rate spikes, delivering oxygen and glucose to your muscles.
Your digestion shuts down to conserve energy, and crucially, your cognition sharpens. Acute stress enhances memory consolidation and sensory acuity. It is a biological masterpiece designed to save your life in a crisis that lasts 3 minutes.
But humans don't just experience 3-minute crises. We experience 30-year mortgages, chronic relationship anxiety, and a relentless 24-hour news cycle. We shift to the far right side of the inverted U curve.
We enter the realm of chronic, sustained stress. And baiting the human brain and glucocorticoids for weeks, months, or years is profoundly neurotoxic.
To see how it conditions behavior, we have to look at what chronic stress physically does to the architecture we discussed in part 1, the prefrontal cortex and the amygdala. The contrast between what it does to those two regions is staggering.
Let's start with the prefrontal cortex. Our Center for Executive Function, Emotional Regulation, and Long-Term Planning. What happens to it?
Sustained exposure to glucocorticoids literally causes the dendritic spines, the receiving branches of the neurons, in the prefrontal cortex to atrophy and retract. The physical connections between neurons basically wither away. The neural networks become desynchronized.
The hardware is actively degrading.
Rapidly. As a result, working memory fails. You become highly impulsive. Your ability to engage the dorsolateral prefrontal cortex to perform complex moral calculations is severely compromised. Most dangerously, the prefrontal cortex loses its ability to send inhibitory signals down to the amygdala.
The brakes fail. And what is happening to the engine while the brakes are failing?
Chronic stress does the exact opposite to the amygdala. While glucocorticoids cause atrophy in the cortex, they act as a potent fertilizer in the basolateral amygdala.
A fertilizer.
They trigger the release of a protein called BDNF, brain-derived neurotrophic factor. BDNF stimulates massive dendritic growth. The neurons in the fear center of your brain physically expand and forge thousands of new excitable synapses.
So the fear center is entering a state of hypertrophy. It is growing larger, faster, and more connected.
It becomes hyperreactive. The amygdala becomes incredibly efficient at learning new fear associations and simultaneously terrible at unlearning them, a state known as impaired fear extinction. You are locked in a biological state of perpetual vigilance.
And this specific imbalance, a withered prefrontal cortex incapable of regulation, paired with a massive, hypercharged amygdala biases your entire behavioral repertoire toward reactive aggression, anxiety, and profound selfishness.
Which fundamentally alters our capacity for empathy. And the mechanism for this is perfectly demonstrated in the mouse empathy study by Jeffrey Mogul.
I want to dive deep into this methodology because it links the stress response directly back to the US versus them divide.
Right. Mogul's laboratory at McGill University investigates the evolutionary rudiments of empathy. They measure a mouse's pain threshold by observing how quickly it exhibits a writhing reflex when exposed to a mild, noxious stimulus.
Okay, setting a baseline.
The fascinating baseline finding is that a mouse's pain threshold is noticeably lowered when it is placed in proximity to another mouse that is experiencing pain.
It effectively catches the other's pain. It resonates with it.
Yes, an emotional contagion. But Mogul found a strict boundary. This empathic resonance only occurs if the two mice are cage mates, if they are an us. If you place a mouse next to a complete stranger in pain, the pain threshold does not change. There is zero empathy.
Because the brain categorizes the strange mouse as a them.
And what happens when a mammal encounters a them? The sympathetic nervous system perceives a potential threat. Glucocorticoids spike. The stress of the stranger completely overrides the capacity for empathy.
So the researchers manipulated the endocrinology.
They administered a pharmacological agent, like metirapone, which blocks the synthesis and secretion of glucocorticoids. They effectively eliminated the biological stress response.
And the results were profound.
When the stress hormones were blocked, the mice suddenly displayed the exact same level of pain empathy for the complete stranger as they did for their own cage mate.
By chemically turning off the fear response, they vastly expanded the mouse's circle of empathy.
And this translates perfectly to human neurobiology. Researchers have replicated this dynamic in human subjects. When humans interact with strangers, their pain empathy measured by observing the activation of their own pain networks in an fMRI is significantly blunted compared to when they watch a friend suffer.
But there's a way to reverse it, right?
Yes. If you administer a drug that blocks glucocorticoids, or even if you just have the strangers play a cooperative video game like Rock Band for 15 minutes prior to the experiment to naturally lower their stress, the empathy returns. Stress, on a cellular level, shrinks our circle of us. It forces our neurobiology to build walls.
So if we establish that stress hormones shrink our empathy, and oxytocin weaponizes our loyalty based entirely on whether someone is in the in-group or the out-group, the inevitable biological question is, how does the brain draw the line between us and them in the first place?
That brings us to part 3, the neurobiology of complex social behavior and moral choice.
Exactly. To comprehend the depth of our tribalism, we first have to respect the sheer, terrifying speed at which the brain categorizes the world. We like to believe that prejudice is a conscious, learned, cognitive construct. But neuroimaging reveals that the initial sorting process happens entirely beneath conscious awareness.
We are talking about fractions of a second. 50 milliseconds.
50 milliseconds.
Yes, in fMRI studies, researchers flash images of faces to subjects for just 50 milliseconds, a duration so short that the visual cortex barely registers the image, rendering it subliminal. If the face belongs to someone of another race or a defined outgroup, the amygdala activates almost instantly.
Before the subject even knows they saw a face, their threat circuitry is firing.
Simultaneously, the fusiform face area, the specialized region of the cortex dedicated to recognizing individual, distinct human faces, fails to activate as strongly as it does for a same-race face.
Wait, so the brain actually processes them as less of an individual?
In less than a tenth of a second, your brain has flagged the individual as a potential threat and fundamentally refused to process them as a distinct, individuated human being.
But the threat response is only half the equation. A-them doesn't just evoke fear. Throughout human history, outgroups haven't just been feared. They have been treated as biologically repulsive. And this takes us to the architecture of the insular cortex, which might be the most fascinating evolutionary hijack in the entire brain.
The insular cortex, or the insula, is an honorary extension of the prefrontal cortex. In all mammals, its primary ancient function is processing gustatory and olfactory disgust. It is a vital defense mechanism against pathogens.
Like preventing you from eating something toxic.
Right. If a rat bites into a piece of rancid, rotting meat, the insula lights up, triggering an immediate involuntary reflex to spit it out, gag, and vomit. It protects the organism from being poisoned.
But human evolution requires immense social complexity. We need a way to process social norm violations. But evolution doesn't have the luxury of inventing a brand new brain region from scratch. It's a tinkerer. It repurposes existing hardware.
Evolution hijacked the insula. Over millions of years, the human brain expanded the insula's portfolio. So now, if you accidentally bite into a spoiled piece of food, your insula activates to initiate the gog reflex. But if you simply think about a horrific moral violation, a politician taking a bribe, an act of horrific cruelty against an animal, or a deep personal betrayal, your insula activates in the exact same neurobiological manner.
The abstract moral violation is processed as a literal physical pathogen. The metaphorical becomes visceral.
This is why our language reflects this biology. We say a bad action leaves a bad taste in our mouth. We say a corrupt institution makes us sick to our stomach. We are describing the literal activation of the insular cortex.
But this biological hijack becomes profoundly dangerous when we apply it not just to abstract moral concepts, but to specific groups of people.
Right. The history of the Cagots of France illustrates this better than almost any other example. Let's delve into how a society can invent them out of absolutely nothing.
Yeah, the Cagots are a devastating historical puzzle. They were a persecuted minority group living primarily in western France and northern Spain. Their severe marginalization began in the 11th century and persisted relentlessly for nearly a millennium.
1,000 years of persecution.
Yes, they were forced to live in segregated ghettos on the outskirts of villages. They were legally required to wear a goose foot pinned to their clothing to identify themselves. They were forbidden from touching public fountains, forced to use separate, humiliatingly low doors to enter churches, and restricted to trades like carpentry or grave digging.
And the majority population viewed them with a profound, visceral disgust, treating them as a diseased, leprous underclass.
And the most incredible part of this massive, centuries-long apartheid is the biological reality of the Cagots. They weren't physically different at all.
The reality is that there was absolutely no objective difference between the Cagots and the majority population. They did not have a different skin color, they did not practice a different religion, they did not speak a different language, and they did not even have different regional accents.
They were genetically and physically indistinguishable from the people persecuting them. So how did it even start?
Historians still debate the origins. Perhaps they were descendants of Moorish soldiers left behind after a retreat, or early Christians persecuted by non-Christians. The initial trigger was entirely lost to time, yet the persecution self-sustained for generations.
The majority population learned to feel genuine biological disgust toward an arbitrary label.
It proves conclusively that dividing the world into us and them is not a rational process based on empirical differences. It is an effective brain state. We unconsciously decide someone is a them, our insula generates the disgust, and then we utilize the immense computing power of our prefrontal cortex to backfill the intellectual justification for our bigotry.
And we can map this exact process in modern neuroimaging using Susan Fisk's warmth competence grid, right? How does the brain sort society today?
Fisk, a brilliant social psychologist, discovered that humans universally categorize other groups along two primary axes. warmth, and competence. Warmth asks, are the intentions of this group friendly or hostile? Competence asks, does this group have the societal power and capability to carry out those intentions?
This creates 4 distinct quadrants of societal perception.
Exactly. Groups perceived as high warmth and high competence are your us. You feel pride toward them. The medial prefrontal cortex activates heavily, viewing them as complex humans. Groups with high competence but low warmth, say, a wealthy minority group or a rival corporate class, evoke envy.
And high warmth, but low competence.
Groups like the elderly or the mentally disabled, they evoke pity.
But the darkest quadrant is low warmth and low competence.
When subjects in an fMRI are shown pictures of individuals typically relegated to the low warmth, low competence quadrant, such as drug addicts or the unhoused, the neural signature changes terrifyingly. The medial prefrontal cortex, the Center for Humanization and Theory of Mind, practically shuts off. Instead, the amygdala and the insular cortex light up.
The brain is actively refusing to process the image as a fellow human being. It is processing the image using the exact same neurobiology it uses to process a pile of rotting garbage.
It is the literal neurobiology of dehumanization.
Which leads to a fascinating, if unsettling, implication. If our highest, most complex moral judgments and prejudices are physically tethered to the ancient biology of our gut, to the insula-generating nausea, could manipulating our physical state alter our morality? Could taking an anti-nausea pill technically make us more tolerant of bad behavior?
It sounds absurd, but the data actually supports the underlying premise. While an anti-nausea pill isn't a cure for systemic prejudice, studies manipulating visceral physical sensations demonstrate clear downstream effects on moral judgment.
Give me an example of that.
For instance, if you have subjects physically wash their hands with soap, or if you simply place them in a room that smells faintly of citrus cleaning products, they become significantly less harsh in their condemnation of moral transgressions.
The physical sensation of cleanliness and purity bleeds directly into their psychological assessment of moral purity.
The bidirectional link is undeniable. Our cognitive morality is heavily anchored by our visceral biology, which seamlessly brings us to the final critical mechanism of social behavior, the biology of empathy. If the insula drives disgust and separation, how does the brain actually bridge the gap and feel another's pain?
And more importantly, how does it decide to take action to alleviate it?
Yeah.
The engine of empathy is located in the anterior cingulate cortex, the ACC.
To understand the ACC, you have to break pain down into 2 components. If I accidentally burn your hand on a stove, your somatosensory cortex maps the exact location and intensity of the burn. It tells you, my left index finger is experiencing high heat, but it's the ACC that provides the negative emotional valence.
The ACC is what screams, this is agonizing, I hate this, make it stop.
Exactly. So the evolutionary leap for empathy was to untether the ACC from direct physical stimulation.
Untethering it so we can feel it without being physically hurt ourselves.
Precisely. If you are sitting in an fMRI scanner and you simply watch someone else get their hand burned, your somatosensory cortex stays quiet. Your hand isn't physically burning, but your ACC activates. Your brain utilizes the exact same neural circuitry to process the emotional horror of someone else's suffering as it uses to process your own physical agony.
On a neurobiological level, your pain literally becomes my pain.
But this shared neural architecture creates a profound biological vulnerability. We call it the danger of intense empathy. Empathy is universally praised as a moral good, but it is not always a reliable engine for moral action.
Why not?
Because if you witness someone in terrible distress and your ACC fires up strongly, you experience what neuroscientists call as if pain. It feels as if the tragedy is happening directly to you.
And if you lack the cognitive regulation to manage that, your nervous system just panics.
Exactly. If the vicarious pain causes massive sympathetic nervous system arousal, if your heart rate spikes, your blood pressure surges, and your amygdala goes into overdrive, you become overwhelmed by anxiety. You enter a state of personal distress.
And a biological organism in profound distress only has one imperative:
to alleviate its own suffering. And if you are overwhelmed by the pain of watching someone else suffer, the fastest, most biologically efficient way to reduce your own distress is to simply look away.
To ignore the victim.
Yes. To walk past the unhoused person on the street. To change the channel when the news shows footage of a war zone. Intense, unregulated, passionate empathy often paralyzes us. It leads to profound inaction because the personal emotional cost of engaging is simply too high.
So how do we transcend that biological paralysis. How do we transition from feeling bad to actually doing good? How do we move from raw empathy to actionable compassion?
The neurobiological distinction between empathy and compassion is perfectly captured in the fMRI studies of Mathieu Ricard. Ricard was a highly trained French molecular biologist who left his career to become a Buddhist monk. He is an expert in traditional contemplative practices.
So they put him in a scanner?
Scientists placed Ricard in a scanner and asked him to engage in a state of intense, focused empathy, to meditate deeply on the profound suffering of humanity.
But what did the scanner reveal?
His ACC and his amygdala lit up with massive intensity, just like the circuitry of anyone else experiencing as if pain. But Ricard quickly reported that the state was emotionally intolerable. He was experiencing empathic burnout. The shared distress was exhausting and paralyzing.
So what did they do?
The researchers asked him to switch his cognitive state. They asked him to engage in the traditional Buddhist meditative practice of compassion.
How does the cognitive framework of compassion differ from empathy?
Empathy is the immersive sharing of the pain. Compassion is the outward-directed, warm desire to alleviate that pain. It is a subtle cognitive shift away from absorbing the distress towards strategizing a solution.
And that subtle shift completely rewired his brain in real time.
The neurobiological transformation was absolute. When he shifted to compassion, his amygdala quieted down. The anxious, as-if pain circuitry in the ACC drastically decreased. Instead, the fMRI showed massive activation in two new areas: the dorsolateral prefrontal cortex, which is responsible for cognitive control and executive planning, and the mesolimbic dopamine system.
The mesolimbic dopamine system, the brain's fundamental reward pathway.
Yes. Feeling compassion for suffering people activated his brain's reward center. Because compassion, unlike raw empathy, is a proactive, regulated state. It is the prefrontal cortex asserting control, saying, I recognize your immense pain. I am not going to let it destroy my nervous system. Instead, I'm going to formulate an effective plan to help you.
And executing a pro-social, goal-directed plan releases dopamine. It is biologically rewarding to be effective.
That completely redefines how we should approach moral action in society. We spend so much time trying to make people feel more, but passionate shared distress is biologically inferior to dispassionate, effective kindness.
Feeling the pain is just the sensory trigger. If we don't engage the prefrontal cortex to regulate that emotion and convert it into directed action, the empathy is useless. It becomes nothing more than an indulgent emotional exercise.
And that profound transition from the automatic, deeply ancient circuits of pain and fear up to the highly evolved regulatory power of the prefrontal cortex is the ultimate synthesis of Sapolsky's Behave.
If we step back and view this massive multi-part journey as a single mosaic, what is the final conclusion we can draw about human nature?
The conclusion is that human behavior is unimaginably beautifully complex. It is scientifically impossible to conclude that any behavior, whether it is an act of horrific violence or an act of profound altruism, is caused by just a single gene or just a hormone or just a childhood trauma.
It's all connected.
Every single action is the end product of an unbroken chain of biological and environmental influences. The sensory triggers of the previous seconds, the hormonal baths of the previous days, the neuroplasticity shaped by months of chronic stress, all the way back to the evolutionary pressures of our ancestors.
But that doesn't mean we are helpless.
Exactly the opposite, because the brain is profoundly plastic. It physically rewires itself based on experience, learning, and cognitive effort. By understanding the granular biological mechanisms behind our absolute worst behaviors, by recognizing how stress shrinks our empathy, how oxytocin fuels our tribalism, how our insulin manufactures disgust, we acquire the exact tools needed to intervene.
We can construct environments that reward fairness instead of aggression. We can use our prefrontal cortex to override our amygdala.
We are not prisoners of our biology. We are active, conscious participants in it.
We've traveled from the split-second firing of a neuron back to the invisible smells and sensory shortcuts of the minutes prior, back to the hormonal amplifiers of the days before, all the way out to the arbitrary societal constructs of us versus them. We are the sum of all these moments.
And the architecture is always capable of change.
So we leave you, the listener, with a final thought to mull over. A question about the very nature of your identity. If our deepest moral judgments and our most aggressive impulses are this exquisitely malleable, capable of being entirely shifted by an invisible smell in a room, the stress of a bad day at work, the invisible spike of a hormone, or the subtle shape of a stranger's face, how much of your core personality is truly a fixed, enduring trait? And how much of you is just the sum of your recent biology? The next time you feel a flash of absolute righteous certainty about someone else's character or an intense wave of disgust toward a group of people, pause. Ask yourself, is that you talking? Or is that just your insular cortex?